just characterize OA, ocean acidification, as a complex multi-level problem due to changes in water chemistry that affect the carbonate biomineralogy, but with multiple levels of consequences, from molecular to cellular, individuals and populations, ecosystems, and all of this can best be considered in the context of space, time, and natural variability, which of course is the milieu in which evolution occurs. One way of looking at this collection of cells or cubes, I can also give credit to Hannes Bowman for creating this for us, and I think Hannes, if you're online, I think his ideas were inspired by his child's building blocks he saw one day on the floor. But this collection of blocks shows that there are multiple interrelated levels of effects of ocean acidification. I think each of us can find, or those of us who do research can find our roles at some level of these cubes, or perhaps multiple levels of these cubes. But how do we approach this complex multi-level problem? Well, one research strategy I've summarized here, and I think it's sort of typical of how one goes about understanding a new problem, and I think OA in many ways is a new problem that, as I said at the outset, not only identifies a number of challenges, but also research opportunities for those of us who do research. So one strategy to approaching this problem, and I think this is fairly typical, is that the first level approach, we try to demonstrate the occurrence or monitor, demonstrate the occurrence or the effects of ocean acidification. At a perhaps higher level, we go on to characterize those effects. So rather than bullet one, we're demonstrating effects as the perhaps simple null hypothesis of OA or high CO2 has no effect on outcome. In bullet two, we are interested in the shape, the functional form of that relationship, which allows us to better use that information to predict how responses may occur in future habitats. Building upon the characterization of the effects is to identify the mechanisms. And again, this doesn't have to be done sequentially, and I know some of us specialize in different levels of this, and there's some wonderful work being done on the mechanisms by colleagues in the Southeast. A recent paper by Hira and Grossell and on the acid-base relationships in fish is a good example, and also that leads to the molecular and suborganismal neurosensory responses. Similarly, Browner out in Hamilton out in the Pacific Northwest has done some excellent work on the mechanisms with respect to ocean acidification in behavior, growth, metabolism, olfaction, and so on in salmon. So a variety of people are working on that level and doing some really exceptional work, which I highly encourage you to check into. Beyond the identification of the mechanisms, and this is important, is how do we go about determining the consequences of these effects? How does it affect the life history, ecology, population dynamics, or the ecosystems that these animals live within? And lastly, to try to tie this all together, it's important to have a handle to allow us to at least estimate the likelihood of these occurrences and effects taking place. So again, I think this slide summarizes a wide range of opportunities and challenges that each of us can find ourselves within, and I want to stress from my perspective at least, one of the really great aspects of the ocean acidification research front is it brings together scientists from a variety of disciplines and identifies joint problems that each of us bring our own skill set to, and they have fostered, at least in our case here, and I know elsewhere, some very wonderful collaborations. So I hope that this presentation, if anyone is interested, do not hesitate at all to contact me or my colleagues about possible future collaborations. Okay, in terms of the occurrence of high CO2, this is a plot that's shown often, but I think it's a very good plot because it does show without much ambiguity at all that the atmospheric CO2 has been increasing over the last 70 to 80 years, and certainly since industrialization. The level of CO2 in the seawater has also increased, as shown here in the blue, with obviously some interannual variation. And the pH of the seawater has decreased over this time period. And these are all readings taken at a fairly isolated location, meaning away from shores in Hawaii. So this increase in CO2 and lower pH, less carbonate, may, and an underlying emphasis, may affect a variety of biological outcomes, including deposition of shells, exoskeletons, fish otoliths, ecological life history rates, viability, growth, etc., and the well-being, health, or condition of the organism. There are examples, really nice examples, of show the effects of high CO2 on shell deposition and dissolution, whether it be in pteropods, corals, a variety of urchin species, etc., have a much broader literature than is currently available with regards to the fish literature in the context of OA. But there are now is now a rapidly growing literature on effects of fish, and many of those papers have been on tropical species, especially off the Great Barrier Reef in northeast Australia, that have shown a variety of subtle and not so subtle effects, but many sublethal effects that were unanticipated when the work began. And we too here in the northeast and in the higher latitude temporal and boreal habitats are seeing effects on fish species of our regions, including the embryo viability, larval development and size, as well as late larval features such as metamorphosis. One way we're approaching the problem in terms of what are we doing about it is we are undertaking a broad brush analysis of what's been published through a meta-analysis, where meta-analysis is simply the quantitative analysis of data from other sources that allows us through statistical methods to basically compare apples and oranges and also test hypotheses about the design, organisms, habitat, phylogeny, and any association that might help us explain the patterns that we're seeing. So along with several interns, we've been collecting such data and analyzing them to look at general trends in the fish early life stage responses to high CO2. And these are from laboratory experiments. Just to give you a sense of where these studies that we've collected have been done, from this map of the world, where the fish icons are located is the laboratories where these studies have been done. And I think your first reaction probably is that, not at all homogeneous nor random. It's fairly strongly concentrated in a small number of labs in the northern hemisphere and some in northeast Australia. Similarly, if you look at the collection literature, it's growing rapidly, and it continues to go. It's about double. That 2013 that was the data as of mid-year in 2013. That bar extends to twice that height, and 14 is even a larger number of papers. But these papers are again non-random in the sense that it shows the publications by country of origin that we use in this analysis. In this case, for this analysis, it was 18 publications. But many publications have more than one experiment reported within them, and so this next pie diagram shows the percentage of experiments by country, and there were a total of 72 experiments that we included in our analysis and with attribution to the country of origin. And lastly, just to give you a sense of the coverage of data, here's the attribution to the taxonomy of these species that were covered in these studies. And I won't spend much time on this part of our work today, but I do want to give you a sense of some of the results. And this kind of study in general is displayed as a what's called a log response ratio, where variations that depart from zero, either above or below, are considered to be significant. And as those that do this kind of work realize that not every study makes observations on the same responses. So this set of 6 responses were those that had a large enough sample size for us to conduct these analyses. And what we found, and to summarize them quickly, that there was a significant effect of high CO2 in these experiments on larval survival, larval size, at least the mass, olfactory capabilities, and otolith size, specifically otolith length, with the N as you see on the slide. It's worth noting that the sample size again varies across different response variables, again, because not everyone observes the same responses in their study. The pattern in terms of attribution to different features that these animals either come from or their life history style, we found that otolith size varied significantly between taxonomic families. This is otolith size as a function of the CO2 level, not just otolith size, period. But the CO2 effects varied with phylogeny, and also varied with the type of egg the fish laid. Was it a pelagic spawner with buoyant eggs, or a fish that spawns demersal, negatively buoyant eggs that subsist and incubate on the bottom of their respective habitats? And as I mentioned in the pie diagrams, there are biases in these data with respect to the phylogeny and also the geographic coverage. So our sample size now is up to around over 100 studies, and these patterns remain consistent in terms of the ones you see here, and we'll elaborate on that in our publication on this topic. So the way we've been spending most of our efforts and funding and time is through experimental approaches to understanding how OA affects biological responses. And the way we approach this is through a strategic stepwise approach. Step one is a one-way experimental design where we manipulate CO2 level and measure a response. And the responses, I'll get into them in detail shortly, but say for instance, survival of the embryos. And then this is a hypothetical drawing in this case, but we look for the shape of that response. And then if, again, depending on the laboratory, its capabilities, the urgency, the question to be addressed, one may either take a subsequent step 2 or jump immediately to a step 2 approach if they have that capability, and look at the cross effects of CO2 with another factor. And here, again, hypothetical plot, but plotted it versus temperature. Others are looking at dissolved oxygen, contaminants, a variety of other environmental factors that could interact with the effects of CO2. And here we see this hypothetical data with the green strips show that the response, and again, let's assume that's survival, decreases in the high CO2, low temperature sector of the experimental space. That may lead one to, in the next go-around, or perhaps another lab, to identify and evaluate that more finely parsed grid of CO2 by temperature to evaluate the functional form of that response in that part of the experimental space. But in reality, most studies deal, whether it be with one factor CO2 or two factors, and I don't know anyone who's looked at three factors, using for fish at least. Typically, there are 3, maybe 4. I can't recall any study that has looked at 5 levels of a response. Now, this is important for those of us who are interested in trying to capture and characterize the functional form of the responses. Certainly with 2 groups, or even with 3 groups, one can test the null hypothesis of no effect of CO2 on response. But to have a robust, tight characterization of that algebraic or functional form of the relationship requires 3+ levels of CO2 to be used. And in the evolutionary literature, a phrase that also often comes up with respect to other environmental variables that perhaps are more easily manipulated than CO2. Let's say, for instance, temperature is the reaction norm. How did 2 genotypes respond? How does one genotype respond across a range of different environmental features, say multiple temperatures, or in this case multiple CO2 levels? And that functional reform gives the investigator a sense of the plasticity of that organism, its ability to respond to, acclimate to perhaps the environmental challenge that's facing it. It doesn't say anything necessarily about the potential to adapt, because that has to bring into play the underlying genetic basis of that relationship. So let's go specifically to the experiments we've been doing. We are at Sandy Hook, New Jersey. The arrow shows our location right at the northernmost point of the Jersey Shore. And we have been evaluating the effects of high CO2 on 2 species of flatfish, summer flounder, or known locally here as fluke as well, spawns in the autumn in the offshore continental shelf waters and releases pelagic eggs. The early life history habitat, specifically the embryos and larvae, exists in the autumn winter waters over the shelf, and this species is one that spans the geographic region from the Mid-Atlantic Bight to the South Atlantic Bight. So for those of you online who are specifically interested in the Southeast and fish fauna and its effects on CO2 on those fauna in that region, this species is relevant. I'll also point out, by the way, that the only other flatfish besides our work on winter flounder that I know of, if I'm recalling at least, is Paracletes olivaceus, which is the Japanese flounder. So it's a congener, close relative of summer flounder, and the patterns that have been reported by Kim et al. Are similar to what we report here. I'll go over those in a minute. So summer flounder is one of our 2 species that we've been working with. Again, our animals were collected in the inshore waters in the summer before they went offshore to spawn over shelf waters. The second species that we've been studying, and we'll make some contrasts between these 2 species, is winter flounder, different family of flatfish, right-eyed flounder, different spawning and early life history habitat. It spawns in winter, inshore, has benthic eggs. The embryos and larvae experience winter waters of estuaries, which are more variable in their water chemistry than the more stable waters that the summer flounder experience. Due to bad luck, I guess, we also have been working with Massachusetts flounder. I say bad luck because we were led to acquire some Massachusetts flounder, winter flounder, due to our lab being compromised by Superstorm Sandy in late 2012, and we missed our local winter flounder season then, so that led us to search more northward of our lab for some fish that we could acquire once we got our lab back up and running. So we have data right now on these 2 populations. I'll be showing some of those in a minute. So to give you a sense of the experiments I'll be going over a summer flounder study in 2012. It's one of the step 1 or one-way designs, 3 CO2 levels, measuring embryo and larval features, survival, growth, development up to 4 weeks post-hatch, which is just about the time they undergo metamorphosis. A subsequent study in the following year, it's a 2-way design, 4 CO2 levels, 3 temperatures, with these responses up to 6 weeks post-hatching. The winter flounder work has been done in 2 experiments. Again, the Massachusetts study, a 4x3 CO2 temperature design, which included, I didn't mention this for summer flounder 2013, but a gamete response, embryos as well as larvae to 5 weeks post-hatching, which is approaching metamorphosis in that species. And the New Jersey study, which is a 4x4 design with the gamete effects as the response variable. I'll get into the details of what we actually measured here shortly. So the hypotheses we've been addressing in these studies that I'll be reporting on today are, 1, are the early life history stages of these 2 flounder species affected by elevated CO2 levels? 2, are the species differentially affected by these environmental factors, CO2 and temperature in some cases, and is there a pattern or association that we can, through speculation or logic, attribute to the reason why they may be expressing different sensitivities to high CO2? And thirdly, are the responses also influenced by interactions with other variables such as the water temperature they're incubated or reared in and the parentage that gave rise to these fish? So in a nutshell, we demonstrate and quantify effects in step 1, species and our population differences in responses are our hypothesis number 2, and the non-additive interactive effects of parentage and temperature in the responses is our type of statistical hypothesis in step 3. The way we've been approaching this is through a laboratory-based experimental system put together primarily by our chemist, Dan Mazurik, with help from other folks for sure, but he's been largely the one responsible for this, and would be glad to answer questions about the specifics of it. Here's a schematic of it. We have 5 temperature troughs, 5 columns in which CO2 is bubbled at different concentrations through this water, and these water at different CO2 levels supply the egg or larval containers in these 5 different temperatures. Again, you saw the designs earlier. We've done 3 level 1-ways and 3x4s and 3x3 designs, so we haven't used all of our capacity, and there's reasons for that. One is just logistics. It would be quite difficult to do a 5x5 with 3 replicas, 75 container study. For anyone who does experiments, you know what I'm talking about. But to give you a sense of how we, what it really looks like, we have a lab under with a loft where we have all the pre-treated water. The PVC pipes is where we actually do the CO2 infusion. The water baths are shown here. They typically are covered with a canopy, which you see in the next slide. Here's one of the troughs where with a constant temperature water bath in which we have the aquarium containers, polystyrene containers wrapped in plastic. This tarp. The inside of the tarp is black. You see that here. Inside of one of the containers with the inflow line, a double-headed outflow line, both mesh covered. The smaller container and the larger container is where we incubate the eggs. We have a floating coffee filter that we use to hold our eggs so we can easily count them and access them, remove dead eggs, and so on. But we release the larvae, and larvae themselves are reared in this larger, 12-liter volume container. So the responses we've been looking at are multiple, and we have various ways we quantify these responses that's shown in this table, and various leads in terms of the institutional lead on these response variables. Furthermore, our experiments are different durations. Like the gamete experiment we'll talk about shortly is very short term. The pre-feeding study would include the embryos and larvae up until the time of yolk sac depletion and starvation. And the longest duration study we've done to date is the entire larval period up through near metamorphosis. We have plans for continuing that onward into juvenile life stages for our next round of funding. Okay, so the summer flounder work, let me go into the specifics of those protocols. Again, our setup, you've seen that. Our CO2 levels, again, this is the one experiment where we had 3 levels of CO2, which give 3 tracks of pHs across a 2-month experiment. Our typical protocol is to collect fish, in this case summer flounder, inshore by pound net when the adults are inshore feeding, hold them in the laboratory, spawn them in the autumn. By spawning, do strip spawning, we know who the parents are, and at the moment of fertilization we can at least track that information for other interpretive purposes. The eggs are incubated in their treatments, either in this mesh basket or in other facilities we have. The larvae hatch, we measure the larvae and monitor them until we terminate the experiment. And for the studies today, I'll be talking about, well, these are the responses we have either measured or preserved the animals for. Today I'll be talking about 4 of the 5 responses shown here for one or the other of the 2 flounder species I mentioned. The summer flounder results can be found in the paper in Biogeoscience, came out last spring in 2014. First, the embryonic period survival. What we found is that at the 3 CO2 levels, there was a precipitous drop in survival to hatching, even at the intermediate level. These levels, by the way, were chosen at that originally because this is as low as we could get the CO2 in our local inshore waters, and these are the levels we felt comfortable with maintaining in our first attempt, and we've improved the control and resolution of the range of values. But I want to point out that most of the responses I'll be mentioning are significant between the low and intermediate levels, and the high level is also, in many cases, most cases, significant as well. There are 3 subgroups that were used to show the control level hatchability or survival to hatching of those 3. I'm showing relative survival to hatching here on the y-axis. So there's a highly significant effect of high CO2, elevated CO2 on survival to hatching. In terms of development of larvae, our typical protocol is to measure the animals at regular intervals, either by calendar or by degree days, depending on the study. We anesthetize live larvae, photograph them, and measure a variety of lengths and depths that reflect both size, and in this case also for a day 0 animal, day 0 post-hatch, the yolks quantity, which is a measure of condition. And more advanced larvae is a 28-day post-hatch summer flounder larva. We measure its body size, length, but also body depth, and other features that are indicative of its condition, its mouth size. One feature shown here in yellow, by the way, is the flexion angles. This animal is in the early stages of metamorphosis. We quantified its developmental state by the radians of the flexion angle. And we, going into this, know that these 10 to 12 measurements we take on larvae are highly correlated. So what we first do with these data is run them through a principal component analysis. And this is what the result of that analysis was for the summer flounder. This is principal component axis number 1, and here's principal component axis number 2. This is for the 28-day-old flounders with these animals in this sort of developmental state. All of these vectors going to the right are basically telling us something what I call the size dimension. So standard length, fork length, body length, total length, mandible length, and this is muscular depth of fin fold and vent. So most of these, the dominant vector in these 2 dimensions is to the right, and I consider this to be a size variable. The one, perhaps to your eyes too, outlier is this vector here, which is clearly mostly a north-south PC2 dimension. And this is a flexion angle, the amount of this bend of the notochord as the animals are undergoing metamorphosis. So I consider this to be a developmental state index. This is a size index for the following plot. So I'll be plotting the next plot. PC1 response, again, as a size, interpret that as size, and PC2 response is developmental state. So this plot shows the PC1 versus CO2 level for age 0, 2 weeks later, 2 weeks old, and 4 weeks old. And what we see is that size actually is smallest at the low CO2 level. Animals are larger at age, at sample, at the intermediate high CO2 levels in the first day 0 and also 2 weeks later. And there's some change in the 4-week response where the highest CO2 level animals are actually smaller than the intermediate CO2 levels. Panel 4 for D here is also at 28 days post-hatch, but this is a PC2 score, so this is our developmental index. The way I scored these, it so happens that the animal that's most developed has the smallest PC2 value. So the high CO2 animals actually are most developmentally advanced, more so than the intermediate and the low CO2 levels. Now for summer flounder, that kind of differential development and size, and again, if you read this as I'm reading this, these animals are going to be nearest metamorphosis at a modest size. These animals are further from metamorphosis and even further still at low CO2 levels, still have growth to do. So I would anticipate if this experiment ran out another 2 to 4 weeks, we'd see a fairly strong relationship between size at settlement, an inverse relationship between size at settlement and increasing CO2 level. For this species, that is important, especially because all the paralictids that I know of, or genus Paralictes at least, spawn offshore and ingress back into nursery habitats as late larvae, transforming larvae, and early juveniles. For summer flounder, they spawn in autumn, again, mostly in September, October, but with some spawning occurring in many other months. But the bulk of the animals are spawned in the fall, and depending on what latitude and when the parents spawn, those animals are going to be experiencing autumn, early autumn, late autumn temperatures. If the animal is developing quickly enough, or the parents spawn early enough, those animals will likely ingress back into the estuaries before winter occurs. Those that aren't developmentally advanced enough for their thyroxine and their sensitivity to thyroxine to be responsive to the thyroxine, those animals basically get locked out due to temperature. Once the temperature drops below 8 degrees, they don't develop much at all. So they're locked out over the shelf waters for the remainder of the winter until the following spring, March, April, whenever the temperature surpasses roughly 8 degrees. So clearly, these effects on development rate and size of settlement are likely to be of ecological relevance to the recruitment of this species. And our colleague Keith Cooper and his students at his lab have also been corroborating these results with respect to the histology and also gene expression of the response of these flounders to these high CO2 levels, where they quantified the craniofacial dimensions, and again it mirrors what we just saw in terms of the size of the animals as a function of CO2 level. The gamete study, and this is done primarily with Ali Cadelmo's lead, who's again a postdoc in our lab, that study first established through preliminary trials an initial objective of finding the milk concentration and flood duration at which fertilization rate began to decline. So any additional stressor like CO2, we're trying to maximize the chance we could pick up that response. So we're looking at that shoulder of change from high to low fertilization rate. And her standard operating procedure for this study was she flooded the milt and collected from the males with treatment water. Again, this is a CO2 by temperature design, so different temperatures and CO2 levels. Flooded the milt for 1 minute, flooded the eggs for 1 minute, mixed the gametes together, and they were in their treatment water for 1 minute, and then rinsed the water off and replaced it with ambient water and scored these eggs for fertilization rate either 24 hours later, then later multiple days after that to confirm the response. And that study had one female each mated with 2 males with 2 or 3 replicates per SIB family. So it was a nested multi-factor design. And what we found here is shown in this plot, somewhat complicated, but fertilization success on the left side with the colored bars representing the means and errors around those fertilization success, with bundled by temperature that the animals were fertilized in. And this on the right side with the blue lines and the blue numbers show the percent decline from 7.9 pH as being the benchmark of ambient conditions. So we see that there's a significant decline in summer flounder with increasing pH level, significant decline in fertilization rate. And here I'm showing this sort of the abstract of the NOAA table with items in bold, even though they're not the only ones that are significant. Temperature had an effect, and so did female in this case. But the ones in bold are those that are relevant to our CO2 and co-stressor interaction. And we found that CO2 had an overall effect. There was a CO2 by temperature interaction, so there's less viability, less fertilization at higher temperatures, and that CO2 also interacted with the female, so there was a suggestion of an interparental difference in tolerance or resilience to CO2 level. And these bullets just summarize what I just said. In contrast to that outcome, the winter flounder showed a different response. This happens to be the Massachusetts winter flounder, and their responses are summarized in this NOAA table, and now I'll go directly to the bullets, where we found that the PCO2 effect occurred, and fertilization increased with increasing PCO2. We also found for the Massachusetts fish that there was a CO2 by temperature interaction with lower fertilization rates at the warmer temperatures for this more northerly population, and there again was a CO2 by female interaction. The New Jersey winter flounder showed something of a similar pattern to winter flounder, the Massachusetts stock as opposed to the summer flounder, in that fertilization rate increased in general with increasing CO2, decreasing pH, increasing acidity, with peak values at intermediate levels. Here's a summary ANOVA results. Here are the bullet points. Again, fertilization peaked at intermediate PCO2 values in the New Jersey winter flounder. There was a significant PCO2 by female interaction. So a lot of data there. Let me just summarize that and with a summary slide for the 2 species. For summer flounder, fertilization rate decreased with increasing CO2 and increasing temperature. It showed evidence of a temperature by CO2 interaction with lower fertilization rates at higher temperatures. Winter flounder increased fertilization rate with PCO2 in both populations. The response to the CO2 varied in terms of the detail with populations and with the environment, the thermal environment. Specifically, the Massachusetts population showed the highest fertilization at highest CO2 levels, and there was a temperature by CO2 interaction. The New Jersey population showed highest fertilization at intermediate CO2 levels, and both species and both winter flounder populations, there was a parental CO2 interaction in terms of differential resilience or sensitivity to high CO2 in terms of just the gamete efficiency and fertilization rate. So what's next? We're completing the analysis of the variety of response variables. I mentioned some already in our table, but they include the variables that we think reflect condition and ecological relevant measures, size, development, and so forth. And our colleague at Rutgers is doing the histology and gene expression. We're also found from what we found previously with winter flounder, our intention is to do a broader interpopulation comparison for winter flounder from distinctly different latitudes and ecosystems. And specifically, we have some data, but we want to expand this to include a more interesting contrast for us, at least, the inshore New Jersey versus inshore Gulf of Maine versus offshore, such as Georgia's or Stellwagen Bank, where clearly the adults are living in a very different environment than the inshore spawning winter flounder are living in. Where the hypothesis is our fish from historically variable CO2 environments, those that spawn inshore, less susceptible to high and variable CO2 predicted for future marine habitats. So that contrast, that test is very much of interest to us and very much in vogue with what's happening on the research front with respect to OA work. So is number 3. We're interested in transgenerational assessment of multiple inshore fish taxa. What that means is almost all, clearly 90% from my scene, of these studies on CO2 effects on fish responses have used offspring and subjected them to a CO2 level or CO2 cross with some stressor. What they haven't done is now being done at some labs, and there's a couple of reports already out on this by Miller et al. In Australia and Bowman et al. In Stony Brook and Yukon, have looked at the effects of the parental experience. Does the adult habitat and CO2 environment influence the offspring's resilience to high CO2? And in that particular case, we have 3 species lined up. We want to evaluate. All are inshore forage fish. And I will say that I did mention this before. Anytime we're doing experiments, and this is true for every lab, there's always going to be a trade-off. You know, what you can handle in terms of volume, treatment combinations, replicates, what fish life stages are important. Fortunately, early life stages are important, and they don't take up much space, which is great. But it's hard to look at adults for this transgenerational study if they require 20-foot diameter tanks to house them. So our focus this next go around is going to be in 3 smaller bodied fish, a gadded, mummy chug, and silverside. All the first 2 of these 3 species, by the way, are found in the southeast as well. Lastly, we're interested in the evidence of heritable variation in the resilience of these fish to elevated CO2 and other co-stressors. And in order to do that, one needs to do a controlled, animal genetics design, quantitative genetics design, where you control parentage and evaluate the heritable versus non-heritable versus other fraction of variance in the phenotypic response due to those different sources. And that will allow us to explicitly look at the potential for the animals to adapt to these future challenges. So in a nutshell, we've laid out in this talk and also in our work a strategic experimental plan which has the intent of casting a broad but efficient inferential net. It intends to cover a large portion of relevant environmental space, looking at species of divergent ecologies that are representative of other taxa, we hope, and measure responses that we think are intended or we think reflect fitness, recognizing that some of these responses are going to be subtle but important in the long run. Our results from summer flounder show clearly a high mortality and increase in CO2 levels for the embryos, and effect on larval body size and developmental rate. And these are effects on genetic rate or development clearly have a relationship to habitat use and recruitment in nature for this species. Our results from the gamete experiment show these 2 species do respond quite differently to high CO2 levels, and even the populations of winter flounder respond differently from each other. All show evidence of a parental by CO2 interaction. And lastly, our meta-analysis, taking advantage of a growing worldwide literature, shows there are significant effects on these responses: survival mass, cell size, and olfactory capabilities. By the way, this is not true for every study, but the sample size is large enough that one can draw probabilistic conclusions based on the analysis of all studies. And they also offer us a chance to evaluate whether or not these differences are due to taxonomy, life history, and habitat, recognizing that there's a geographic and taxonomic bias in the published studies that we look at. So with that, I want to thank my colleagues. This is a group of us, most of the OA team. Beth Phelan isn't in that photograph, but I think everyone else is from 2 summers ago that contributed to much of what you heard about today. And with that, I'll be glad to try to answer any questions you might have.
Great. Thank you so much, Chris. Really interesting and thorough presentation. We will now open the floor for any questions of our presenter. Go ahead and type questions or comments into the questions box in the control panel. As folks are doing that, we do already have a few questions. The first question was in regards to the meta-analysis work. Margaret Miller is curious if you did you control or account for different treatment levels, i.e. concentrations among studies?
Right. Yeah, we did, Margaret. We used a threshold of I believe it was 2,000 parts per million CO2. We did it in both ways, by the way, just to see how robust the conclusions were, and the conclusions were robust regardless of whether that threshold was implemented.
Okay, great. Lee King is wondering if he states that your PCO2 levels were manipulated to 1808 and 4,714 microatmospheres. Considering that these waters are sources of carbon dioxide and are less affected by the increasing atmospheric carbon dioxide levels, when do you think the studied region will reach such high CO2 levels?
Right. 4,000 plus? Never. Maybe in extreme situations, but Lee, you probably are aware of data that show that there's high levels, high magnitude, but also high frequency of CO2 variation in inshore habitats, and that's both in the southeast as well as the northeast and out in the Pacific Northwest, and some of the talks already given in the SOCAN series have shown that. So I think the challenge really is how to, one, fairly characterize the dynamics of the variation in CO2 level and how to implement that in an experimental protocol. And then I think it's also fair to ask if animals in inshore habitats are perhaps more resilient to these same level of CO2 values that we subject the offshore species to. Is that the fair contrast, or should we be dealing with, say, standard deviations of local habitat variations? So that gives us a, how many standard deviations above what they're currently existing in would elicit these kinds of responses. So there are a lot of nuances there in the actual treatment implementation that need to be recognized.
Great, thanks. There was a question, you mentioned a publication early on in the talk about identifying OA mechanisms from some folks perhaps here in the Southeast, and Yu Chin Chu is wondering if you have the citation or reference for that paper. I believe it was Gossel?
Yeah, I do.
Yeah.
Wow. Yeah, I have several actually. I'd be glad. You can send me an email, and I'll send it back to you. But there is a review paper by Heyer and Grossell that I referred to. I think it was an annual review of. Let me. I don't want to lead you down the wrong path. Shoot me an email, and I'll respond to that with the citation.
Great, awesome. Peter wants to thank you for the very interesting presentation, and wonders if you see effects of a higher CO2 level directly on the development of the skeleton. For example, a later ossification and or weaker ossification?
Yeah, Peter, I can't, at this point I can't say because we haven't reared the animals till later, okay? One aspect of your question perhaps that we're still dealing with is to what degree is the pattern of the craniofacial shape change due to normal metamorphosis. They're going through the changes that the whole animal's going through versus aberrant, if you will, dimensions of these bones. We haven't done that residual analysis yet. That's certainly something that we want to pursue further in terms of how these might affect the animal in terms of latent response. This current round of funding that we're operating in and other proposals we have in review right now, we hope to be able to evaluate winter flounder through their first summer of growth. So we'll have animals that are on the order of, sorry, winter flounder, I'm not sure if I said that correctly, but on the order of 4 to 5 centimeters by the end of the study. So hopefully that kind of age will allow us to evaluate those latent responses.
Great. We have a question from Ann Faria. She again, or he again, is thanking you for a great presentation and would like to know your opinion on one topic of lab experiments on OA. The levels of PCO2 vary greatly in the literature, and it's very difficult to take any conclusions regarding which species are more or less vulnerable, which responses, behavioral or developmental, are more or less conclusive. The PCO2 levels likely to occur in an open ocean environment are certainly different from levels likely to occur in coastal systems. How do we standardize this? My opinion is that we should use levels predicted to occur in real habitats.
Right. That's a long question. A lot of typing there. What I would say is, for our meta-analysis, what we're doing is putting the responses on a common dimension, the CO2 levels, so we can identify, the overlap or non-overlap or the range of values used, and bundle those by their source habitats. So we are making efforts to attribute those patterns to realistic contrast with what they're experiencing in nature. So I will say there are significant patterns, and I don't think what we're showing and others have shown are artifacts of the experiment. There are robust, repeatable patterns in what we're seeing.
Great, thank you. Isat is wondering, does parents, particularly the mother, age or egg size impact carbon dioxide sensitivity of larvae?
Okay, well, good question. I'd love to answer that question too, maybe with some more research. I'm very much interested in maternal effects and reproductive effort and so on. We can't answer that yet with what we've done. Yeah, just to be brief about it, to give you a quick answer, in order to address some of those issues, we would have to maintain the SIB groups separately, and usually we pool them in our studies, so we don't have the resolution to determine that. In our future studies, our proposals include the implementation plan to track individuals by microsatellite, and therefore be able to extract their family heritage even after they've been pooled in groups. So we hope to be able to get to that. But those are attributes that may be important, and if you do experiments, you appreciate this, to add another layer, either another co-stressor or a family design is a good example. Can tremendously increase the actual treatment combinations and scope of the study. So you have to be really careful and thoughtful about how to maximize what you're getting for the effort. Where you're tapped out, where you can't go any further.
Thank you, Chris. So we have our next question all the way from Colombia. Thanking you for your presentation. And Marcella is wondering how you interpret high fecundity responses.
High fecundity responses, I'm not sure what that means.
Let's go ahead and see if he follows up. We'll see. Marcella, if you could follow up on your question in the questions box, that would be great, and we'll ask another one in the meantime. Ben is curious if there is any indication of acclimatization by different species to OA levels. Is anyone studying this?
Yeah, I can't give you a citation. I'm sure that I'm sure they are. Probably more so with inverts than fish. Yeah, I'd have to get back to you on that, Ben. Sorry.
Okay, great. So Marcella asks or furthers his question. He's wondering if there could be different interpretation with those high fecundities being seen. Probably getting at other factors.
Right, I'm still. Again, maybe Marcella, if you could email me and we could go back and forth on this. We're still not sure what you mean by high fecundities. The species we work with, the two flounders, are highly fecund marine species. The female effect I was talking about in the ANOVAs and the female by CO2 interaction is the identity of the female. The females their offspring have different responses to the CO2 levels they're exposed to. That was the fertilization study.
Aha. Okay. So Marcella, if you wouldn't mind, Chris Chris's email will be made available in the chat box on the side, and so feel free to follow up with him so we can make sure that that question gets answered. We will, if there are any other questions lingering out there, just wait another moment or two for anyone. There was great questions so far. All right. So I don't see any more coming in at this point. But there are a lot of thank yous. It looks like some students were tuning in as well, and they were able to learn quite a bit. So thank you. Well, all right. So we will go ahead and once again, if you have any questions that we didn't get to and would like them answered, please feel free to email Chris. That contact information is in the right-hand side of your control panel in the chat box. If you're interested in joining the SOCAN listserv to learn about the Acidification Network or receive updates in the future, please sign up at the URL shown here, the socorro.org/SOCAN. And you can direct any questions about the Southeast Ocean and Coastal Acidification Network to socan@socorro.org. Thank you for attending the SOCAN State of the Science webinar today. We always welcome feedback, further questions, or suggestions for topics moving forward in this series. And you can submit input simply by replying to the follow-up email you'll receive tomorrow or emailing that SOCAN email address. The recording of this webinar and a PDF of the presentation will be available on the SOCAN website. And we do have our next State of the Science conversation coming up in 2 weeks on May 5th. Leslie Wicks and Peter Etnoyer of the NOAA National Centers for Coastal Ocean Science will be discussing cold water corals with a talk entitled Crumbling Coral, Cold Water Reefs in the Northeast Pacific and Their Implications for Other Regions of the USA. So with that, we hope you will join us and want to thank you again, Chris, for a really informative presentation. Thanks to all of you for your thoughtful questions and taking the time to join us. And we hope to see you at SOCAN's next State of the Science conversation. This is the end of the webinar.
Thanks, Jim.
Thank you.